Good morning, students. In last lecture we studied about the natural method of vegetative reproduction. Today we want to study about the artificial method of vegetative reproduction. Artificial method is nothing but the man-made activities. So in this artificial method mostly done in horticultural plants. Horticultural plants is nothing but the fruit plants. It have the different methods: cutting, budding, grafting, layering. All these methods are done in rainy season, spring season, mostly done in August to September. It have the favorable condition, all over cold atmosphere. So from starting, first one to study the aims of the methods are: first one is rapid multiplication. Second, combining of desirable characters together. These methods are cutting, grafting, budding and layering. First we want to study about the cutting methods. It is most simple and rapid method. Cutting gives better result than other methods. Plant produced by these methods are generally similar to parent plants. It has some types, that is the stem cutting. Stem cutting is nothing but take the stem of 10 to 15 centimeter length with one or few healthy buds is selected. Second one is slanted cut is given in the base of stem. Due to maximum cambium, tissue is exposed. Next, rooted promoting hormones like IAA, IBA, NAA. These are the hormones, that is the indole acetic acid, indole butyric acid, are applied to the cut end of stem. Next, spring season is most suitable for cutting. Stem cutting of herbaceous plant stem, non-woody, green is called green cutting. Example, stem cuttings are mostly done in rose, sugarcane, China rose, bougainvillea, etc. The root cutting, next one is root cutting. In this root cutting, on the surface of the root are present adventitious buds. Next, 2 to 10 centimeter part of the root is used for cutting. Then, new root and shoots develop from the same place. Examples are lemon, apple, tamarind, raspberry. Then in lip cutting it is carried in thick leaves, fleshy leaves. Example, bignonia, calonici, bryophyllum. That is nothing but at the margin of the leaf are present many foliar buds. Next one is budding. After the cutting, budding method is done. Budding, it is one type of artificial vegetative reproduction used by gardeners. In this method, a bud with small portion of surrounding bark is taken from the stem. Then this bud is inserted into small slit and made the bark of stalk. Then bud and stalk are tightly tied together, but bud portion should remain exposed outside the air. Both are kept moist by sprinkling water on it. Slowly, the tissue of buds and stalk joint, and from buds a new plant is produced, and it is mostly done in rose, apple, peach, etc. After budding, the grafting method is done. Grafting mostly done in fruit plants. It is an organic union of two related or compatible plants which have vascular cambium. It is not applicable to monocots because cambium is absent. Second one is basal part which provide root system is called stalk or rootstock or understock. Next, upper part which provides shoot system is called scion or graft. Next, slanted cut is given at lower end of the scion and similar cut is given at the upper end of the rootstock. Then, union of two slanted cut, that is the graft, is made in such a way that cambium of two plants come in direct contact with each other. Grafted region is covered with grafted wax. It walls avoid drying and infection of germs. When buds on the scion sprout, then all the buds and branches of rootstock are cut off, otherwise they will not allow the growth of scion. Scion selected is of superior quality than rootstock. This method is useful in weak rooted plants. Example, scion of the Seri mango is grafted on the stalk of Desi mango. Example, apple, citrus, pear, peach, etc. So in this grafting there are different types are there. First one is tongue or whiplash or slice grafting. In this type, diameter of stalk and scion is the same. Tongue grafting is most common and most successful because more cambium tissue of stalk and scion come in contact with each other. Second, wedge grafting. Diameter of stalk and scion is the same. Then crown grafting. Diameter of rootstock is much more than diameter of scion. Next, approach grafting. Scion when grafted with rootstock is still attached to the mother plant. When graft becomes successful, then scion is separated from the parent plant and scion is only allowed to grow on rootstock. Last one is layering. In this method, stem is made to root still. It is attached to parent plant portion of the stem. The stem is slightly injured or notched. This part of the stem is treated with growth hormones and this part is buried below the soil till rooting. After rooting, branches is separated from the present plant and then transplanted. Then air layering or booty. Air stem is slightly injured. This part is covered with grafting clay. This portion is kept moist. After 2-3 months, it shows rooting. Then this branch is separated from the parent plant and transplanted. Then micropropagation. Micropropagation is nothing but the small plant can be enlarged in large number, that is vegetative multiplication of plant. By using tissue or organ of the plant, it is also called tissue culture method. Thank you. Next period we are going to study the next part.